All right, thank you, and thank you for inviting me to give this talk about this, I think, a really interesting discovery. If we think of virus discovery, present days, we usually think about a metagenomic-based virus discovery approach, which has been the gold standard of identifying new viruses, especially in association with pathology. However, metagenomics-based virus discovery is not without its pitfalls, as the figure in here indicates very well. This figure shows the number of each taxa that has been classified by the International Committee on the Taxonomy of Viruses, or ICTV. What you can see is that while in the past decade the number of species and genera have drastically increased due to the wide and fast availability of metagenomics, the same is not really true for higher ranking taxa such as families and orders. And the reason for this is that metagenomics based virus discovery relies on sequence homology. Like if I find a new virus sequence, I can't tell whether it belongs to a virus or not unless I already have something similar to that very sequence deposited in a public database. The information, furthermore, what we obtain this way is limited only to a sequence. We don't really learn much about the virus's potential pathogenesis or what the virus's virion might look like. The question raises itself that whether if we are dealing with a pandemic of an unknown causative agent, is really metagenomics the only way to identify what's behind a pandemic. In this talk, we are going to take an alternative approach through an imaging approach. What you see in here are all electron micrographs. Two of these were taken with negative staining electron microscopy, and one of them by thin sectioning transmission electron microscopy. The first two both come from my six-legged patients. They were common house crickets, and they both were identified because we could not get anywhere by just simply trying to PCR commonly occurring cricket pathogens. So one of these is an iridovirus, and the other one is a densovirus. There is of a drastically different lineage of densoviruses than Acheta domesticus densovirus, which many of you might be familiar with. Whereas the third picture is actually about a human pathogen. This is the original SARS coronavirus, which was also detected by this methodology and associated with the original SARS breakout. So currently, there is a pandemic going on in the United States, but not in humans. This affects actually superworms, and we thought that we could take this approach even one step further. We could use cryo-EM and 3D image reconstruction to identify the causative agent of this pandemic. So the star of this pandemic is the superworm, which is not a worm at all. It's actually the larval form of a darkling beetle species, and it's rare worldwide in large numbers, in masses, as for nutritional purposes of exotic pets, and more so for humans. The superworm also has a close relative, the mealworm, that is reared the very same way. These animals are reared in gigantic masses where they can touch each other so that they don't pupate, and that results in a quite long shelf life. Another darkling beetle species, the buffalo beetle, also commonly occurs in insect rearing facilities. Since 2019, there is anecdotal evidence that there is a superworm shortage all over in the United States, and this superworm shortage is associated with a distinct pathology that results in very high mortality and often complete colony collapses. And this pathology always look the same. These animals, the larvae, at about 8 weeks of age, start erratically wiggling, then they stiffen, and then they gradually blacken and die. So if we look at healthy superworms, then on this piece of cloth, they immediately try to hide, look for shelter, whereas if we look at an affected superworm larva, then this one is not in control of its locomotion anymore. It's trying to hide, but can't really get anywhere. So we were curious what could be going on, and we were especially curious when we were actually contacted by an insect farmer from the state of Utah who suffered a complete colony collapse and an approximately 90% mortality of his superworm stock, and was very frustrated of the entire phenomenon and wanted to get to the bottom of this, so before he restarts he could take the proper measurements to prevent this from ever happening again. So after our contact with him, a couple of days later, we ended up with about 4 pounds of oozing stinky black superworms in our post box at IQB at Rutgers. First of all, to have a look, as I said, we used a cryotransmission electron microscopy or cryo-EM approach. So this methodology involves the visualization of the specimen with an electron beam on an electron microscopy grid that is covered by a thin carbon foil which contains evenly spaced holes. These grids have to make have to be made hydrophilic in order for an even distribution on the sample for them. So we expose it to plasma by glow discharging, and we pipette the sample on top of it, blot away the excess sample containing solvent, and then quickly plunge it into liquid nitrogen cooled liquid ethane, which results in amorphous vitreous eyes forming inside the holes on the carbon layer. And through this vitreous layer, we can drive through an electron beam, which then we can capture with an direct electron detector. So we can take images that contain the multiple projection, two-dimensional projection of our specimen on these micrographs, which we can align and classify, and then ultimately reconstruct a three-dimensional volume, which if our reconstruction is at high resolution enough, and with today's cutting-edge cryo-EM methods, it's quite easy to obtain a high-resolution cryo-EM structure. Then we can even build an atomic protein model into this electron density, modeling what our protein looks like at the protein or molecule looks like at the atomic level. So in order to execute this very same approach with our superworms, we homogenized about 30 blackened superworm larva carcasses and subjected it to a virus purification method on a sucrose step gradient. And this method is frequently used to purify larger molecule assemblies and viruses of potentially all sizes. So we did receive two fractions of this sucrose gradient, which appear to contain protein. So we visualized both of these with our Thales Arctica transmission electron microscope. And what we saw that both of these fractions contained particles at about 28 nanometers in size, which was consistent with these being virus particles. And both of these fractions actually consisted of 4 proteins that were the same size. So if we look at these two micrographs close, what we see is that in the higher buoyancy upper fraction, these are less electron dense than they are in the lower fraction. So this made us think that we weren't dealing with two different viruses in here. This was actually the same virus, but in the higher buoyancy fraction, these were empty capsids, just shells that were not filled with genome, whereas the lower fraction contained the virions that actually packaged genome and were mature. So we reconstructed this 3D volume, which proved our previous hypothesis, since both of these electron density maps, the particles in them have the same surface morphology, yet in the higher fraction they are empty and filled with electron density corresponding with the genome in the lower fraction. So I must emphasize that at this point we had absolutely no sequence information to our deposition, but we wanted to still determine what kind of virus this could be. So how we went about it was that we actually traced the density with just a polyalanine chain, and with this approach, we could reconstruct what the protein backbone looked like in this density. There were 60 copies of this backbone creating this capsid shell. And what we could do with this backbone structure was that we could submit it to a protein structure comparison server called DALI, which what it does is that it takes structural information and compares that to the entire protein data bank, the database where all protein structures that are submitted get deposited. And what the highest scores that DALI gave us were actually all corresponding with the Parvoviridae family, the family that contains parvoviruses and enzoviruses. And out of this family, we got extremely high scores for the 3 enzovirus structures that have been deposited to the PDB thus far, but some minor hits on the over 100 parvovirinae structures or parvovirinae structures as well, the ones infecting vertebrates. So so we compared this backbone structure with the backbone of the capsid protein of the currently existing densovirus structures, and the similarity was really quite obvious because these structures have a very similar fold to each other. But what supported us even more that this is indeed a densovirus was that although this is a completely new capsid surface morphology within the family, it's still a variation of the same topic and completely in line with how other densoviruses look like on their capsid surface, as opposed to other members of the Parvoviridae in the other subfamilies. So now we knew that we were dealing with a densovirus, so it was time to name this new virus, and we named it Zophobas morio black wasting virus, which I will abbreviate as ZMBWV based on its after its characteristic pathology. So what are parvoviruses? Parvoviruses are small DNA viruses which are non-enveloped, which comprise a very diverse family called the Parvoviridae, and this family has just as diverse hosts as the viruses are themselves, infecting such basal invertebrate animals as this beadlit anemone, as well as even us primates and basically all major lineages of vertebrates and invertebrates. This family is united by its single-stranded genome and the very limited coding capacity of this one. They only have one cassette that encodes non-structural proteins that are proteins associated with virus replication, and one that encodes structural proteins, the ones that basically create the viral capsid and are responsible for transmitting the virus between cells and even between hosts. And these genomes are flanked by these partially double-stranded, very characteristic DNA secondary structures. So now that we knew that we were dealing with a densovirus, we also didn't have to spend hundreds of dollars on executing a metagenomic approach and sequence all nucleic acid in our sample. We knew that we have to target a small single-stranded DNA molecule, so we submitted it to a commercial transposase-based sequencing service, and we could sequence the entire genome from $15, which was indeed consistent in its genome content as well as structure with being a densovirus. We could also tell, based on sequence homology searches, that the closest relative to this new virus was Blattella germanica densovirus, which infects the German cockroach. This first surprise, however, came at this point with the sequence that we realized that this virus was actually not alone out there. This virus sequence has already been deposited, at least a very similar one to this, to the gene bank multiple times, but interestingly not from insects, but from vertebrate metagenome assemblies. This came from various bat and bird species, both in Europe and both from Europe and China, as well as from a pangolin assembly from Malaysia. So at this point, we were questioning our discovery that is CMBWV really the pathogenic agent of such a devastating pathology that we can observe if it has been found worldwide before, and we really don't hear worldwide occurring blackening disease in any other insect. So we decided to embark on a journey to study the pathogenesis of ZMBWV in order to confirm whether if it's indeed the causative agent of the black wasting disease of superworms. So first we contacted farms that had the black wasting problem, and we learned from them that although there is an over 90% mortality, there are some superworms that do survive this and manage to pupate, turn into beetles, and complete their life cycle by reproducing. So we collected animals from this farm in all life stages, including even newly hatched larvae, as well as two we called larvae, and we measured the virus titer, quantified the virus titer of ZMBWV in these samples. And what we found that all of these insects had very large, had significantly high viral yield of ZMBWV, which was extremely high in the asymptomatic 8-week old larvae and of course in the blackened carcasses, but was already present at a quite good titer in case of the newly hatched larvae, and even the surviving beetles that were reproducing were carrying this virus at high titers. Next, we administered purified CMBWV virus at 5 different concentrations with direct fat body injections into healthy 4-week-old superworm larvae. And what we found was that all of these started exhibiting the symptoms of the black wasting disease, and eventually all of them died. Only the time of their death depended on a dosage-dependent manner. The ones that got a higher dose died earlier, and the ones that got a lower dose died later. We also administered CMBWV into these superworms by non-invasive methods, including exploiting the natural cannibalistic instinct of the larvae. So we just simply offered them the blackened carcasses for which we knew the virus titer and let them eat these, as well as we dripped the virus suspension at 2 different concentrations directly on their cuticule. And what we got with the feeding experiment, that they again started exhibiting the symptoms of ZMB, of the black wasting disease, and died. Only took them approximately twice as long time to die than it did with the lowest dose in the injection experiment. With the dripping experiment, they also died, and it also took them a significantly longer time. However, it happened as well in a dosage-dependent manner, just like we saw with the injection experiment. Then we repurified the virus from all these dead individuals and sequenced its genome, and what we found that it was absolutely 100% identical to the ZMBWV Utah strain that we infected them with. And halfway to the experiment, on the 15th day, we also assessed the virus titer in these animals, in all of these experiments, where what we found was that actually the lowest virus titer resulted in those individuals who got high dose of injections, whereas the highest virus titer was in the individuals that were administered the virus in the non-invasive way. We also observed another phenomenon, that actually the virus titer increases very suddenly and really significantly, like over 10 times, over 10-fold, once the animal starts exhibiting the symptoms of the black wasting disease. So with these findings, we could fulfill Koch's postulates, and we could establish that yes, CMBWV is indeed the pathogenic agent that causes the black wasting disease in these animals. This sudden titer increase was, however, really intriguing us, as well as another phenomenon, that if we looked at the progression of disease in these larvae, what we saw that there was an area in the abdomen which showed a gradual discoloration until the entire body of the animal blackened eventually, but this area corresponded with where the midgut is located in the animal's body. So what could be the midgut's role in the pathogenesis of ZMBWV? And to find out, we subjected healthy individuals as well as freshly diseased individuals to micro-CT, with which we could render a 3-dimensional rendering of the entire larvae. So this gave us the opportunity to actually look inside the midgut lumen, and what we saw that while in a healthy individual, this midgut looks really what it should look like in an animal that feeds well, with a healthy trophic layer lining the midgut. This was not the case with the freshly diseased individuals. In here, we could only see the remnants of the musculature of the midgut. The columnar cells, the trophic layer of the columnar cells was completely absent, as well as the supporting fat body structures around the midgut, and we observed these large fenestrations on the midgut wall. What we concluded from this is that the sudden increase in virus titer, as well as the manifestations of the symptom, probably happens when this virus, which originally replicates in the midgut trophic layer, breaches the midgut wall after destroying the trophic layer and suddenly invades the entire body of the animal, leading to a systematic infection, and in the result of the onset of this systematic symptoms, as well as a sudden increase in virus titer. So next, what we were curious of that is CMBWV really the pathogen of the nationwide superworm pandemic, or was this just an isolated case that was only true for the Utah farm? So in order to do that, we also turned our attention not only to superworms, but the closely related mealworms, since these animals, apart from being zoologically really closely related, they are also frequently housed at the same farms, and we wanted to get to the end of it, whether the mealworms could have any role in this pandemic, even though we don't hear about mealworm colony collapses as opposed to the superworm ones. So we obtained samples from all over the United States, where all mealworm pools tested positive, as well as all superworm pools, apart from two where the farmers did not observe any signs of the black wasting disease. Our collection of samples made really easy by that we actually released only 1 month after receiving our Utah shipment a preprint at the bioarchive server, which was reached by many who were interested in insect farming and wanted to get to the bottom of this pandemic. So they were more than eager to help and send us samples whenever they dealt with this black wasting pathology of their animals. The mealworms were the second surprise throughout this journey, because when we assessed the viral titer on these mealworms, once we found out that they test positive by PCR for the virus, they actually had the exactly same magnitude of virus titer as our blackened superworm carcasses did. Only these mealworms did not exhibit any symptoms of the black wasting disease. What's more, they were reproducing, eating, just living the mealworm life. And interestingly, when we sequenced the genome in these mealworms, what we found was that the genomes of CMBWV in these animals were actually more similar to those that were previously found in the vertebrate metagenomic assemblies than they were to our North American superworm, virulent superworm samples. This was also supported by the phylogenetic reconstruction, which we did with the complete genomes we obtained, as well as the metagenomic assemblies, which are highlighted in green here. And we found that all of our North American virulent superworm strains actually clustered in one monophyletic lineage, pointing towards a single point of origin, whereas all the mealworm samples or all the mealworm genomes clustered all over in this tree together with the metagenomic assembly originated sequences. There was one superworm pool, however, that is an exception, and at this farm in Florida, they could not observe any sign of the black wasting pathology. So we actually cut these animals as a control by believing that they were healthy. So this was a very cold day in New Jersey when they arrived as a live shipment. And just a couple of days after receiving them, they started to all of a sudden exhibit very suddenly the signs of the black wasting disease. And upon PCR amplifying and sequencing the genome, we could conclude that indeed CMBWV caused its symptoms. So this only pool clustered outside of the North American lineage, but together with the non-virulent mealworm strains and metagenomic strains. So now that we know that this virus is also present in the mealworms, what's more, at very high titers, we were curious that could this be the origin of the pandemic? Could it be that these mealworm viruses are actually virulent in the superworms, and it's a mealworm-specific phenomenon that they do not cause disease? So we repeated our inoculation experiments, but this time our healthy superworms were subjected to either the virulent Utah strain or to a mealworm originated New Jersey non-virulent strain. And what we found in case of fed body injections, that again, the ones that got the Utah strain, they died in a dose-dependent manner. However, the ones that were injected with the mealworm strain, after an initial quick die-off in the first couple of days, also in a dosage-dependent manner of the experiment, they actually kept on surviving. And these animals who survived the initial toxicity were alive for 4 months when we decided to finally euthanize them. In case of the cross-feeding study, where we subjected our healthy superworms to the carcasses infected with the virulent Utah superworm strain, died again at a similar interval than they did in the previous experiment. However, when we fed these mealworm carcasses to the superworm with the exactly same high virus titer, nothing happened. All of these superworms survived. So we could conclude that indeed these mealworm-originated CMBWV strains were indeed non-virulent in the superworms. As I said, the animals who survived the experiment survived for 4 months, so we kept on assessing the virus titer, and what we found that even these 4-month-old animals still exhibited low titers of the mealworm strain of ZMBWV. Next, since now we had strains with 2 different types of virulence, we were asking whether there could be cross-protection achieved by the non-virulent strain against the virulent strains. So we did an experiment where we pre-inoculated our healthy superworms with the non-virulent mealworm strain and let them incubate the virus for approximately 3 weeks. And during this incubation period, as you can see, they they survived. But then when we challenged them with the Utah strain, what we found was that actually those who got both strains did survive. About 35% of that fraction kept on surviving, as opposed to the ones that only got the Utah strain, which died off just like we saw it in the previous experiments. So we concluded that there is not complete, but there is some degree of cross protection, at least between these two strains. So next we were curious that whether all of these non-virulent mealworm strains are capable of providing the same cross protection or even better. And what we found was that it's not necessarily the case. While most of these strains did indeed provide cross protection compared to the single Utah strain inoculated one, there was one strain from Oregon that already caused about 35% mortality in the single inoculation phase, and then after this it facilitated the die-off. So though many of these non-pathogenic strains in mealworms do result in cross-protection, there are some strains that are even mildly virulent also in the superworms. Another phenomenon was that these strains also, even if they provided cross protection, differed in acute toxicity. This, what we see in here, is the percentage of survival after 2 days following inoculation. And what we see that some of these non-virulent strains actually have really high acute toxicity, murdering over 60% of the animals upon inoculation. We were curious what could be the mechanism behind this cross protection, and we first suspected superinfection exclusion. So in order to test that, we pulled insects on a biweekly basis from the experiment following the double challenge, and PCR amplified a variable fraction of this genome and Sanger sequenced it. So 2 days after the challenge with the Utah strain, we can see that both of these genotypes are present in this electrophotogram, whereas only a single genotype is present in the one inoculated only with the mealworm strain. However, at our next sample pool, we could only detect the Utah strain going forward in these samples. It completely eliminated or took over the non-virulent mealworm strains replication. And after the 20th day, when some of the animals started exhibiting symptoms while some still remained protected, we could not observe any difference. It was only the Utah strain found in both animals. So we could exclude superinfection exclusion as the possible mechanism of the cross protection, but the actual mechanism behind this still remains unknown and requires more investigation. You might ask the question whether CMBWV is limited to mealworms and superworms, or does it affect, especially considering that the closest relative to this virus infects the German cockroach, other commonly farmed insects in close proximity. So from one of the farms that suffered the black wasting disease, we received some dubia roach and buffalo beetle samples, buffalo beetle samples, and we could detect the virus in them at quite high titers in all life stages, despite of these animals otherwise also being healthy and not exhibit any signs of the ZMBWV infection. So now that we characterized the pathogenesis of this virus and established that ZMBWV was indeed the agent of the black wasting disease, we could finally turn our attention back to the proteins and the structure itself and see if we can learn more about the pathogenesis of this virus using this data. So first, if you remember the 4 proteins we detected in these fractions, we could identify with tandem mass spectrometry protein sequencing that they did indeed come from ZMBWV. So these capsids are indeed comprised of 4 capsid proteins. We could also finally build the density at an atomic level, and we could find traits associated with this capsid that were really unusual in the entire Parvoviridae family, one of these being a very large fraction of the genome being icosahedrally ordered and interacting closely with the capsid's inner surface in the virions of this virus. As you can see, this region is completely empty in the empty capsids. In here, in fact, one of these capsid subunits, there are 60 of these, interacts at three different regions of the packaged genomes. Genome is basically pinned by the capsid surface at like 3 different locations at each of these subunits. And we also resolved the near atomic structure of 2 other ZMBW strains, one of these being the mildly virulent Oregon mealworm strain and the non-virulent New Jersey mealworm strain. And with this, we could investigate polymorphic regions. These are regions that are single amino acids which are different in these various strains. And what we found that these polymorphic sites were actually all surface exposed on the capsid surface, so they must be associated with capsid-related metabolism of this virus, and that they actually limited into clusters on the capsid surface, one cluster located in this region and the another located on the spikes surrounding the 5-fold axis. And especially this site framed with the red was really interesting to us because this region in Eunonia coenia densovirus, which infects a Lepidopteran moth, was shown to be the region that is responsible for midgut transcytosis. So if we turn our attention to these spikes, they actually form a pocket on the capsid surface, and the polymorphism in here, regardless of virulence, manifests in this pocket being either hydrophobic or being polar. So in case of the hydrophobic instance, as in the Utah strain and in the New Jersey Molitor strain, we can see a small molecule in here, which is probably an N-linked glycan that is associated to this hydrophobic pocket. But in case of the polar pocket, this glycan is either absent or disordered, as we see it in the Oregon Molitor strain. So why is this important information for us for the pathogenesis of ZMBWV? It's important because N-linked glycosylation can only happen on the capsid surface if the capsid passes through the Golgi network when these post-translational modifications happen. So it probably exits the cell through a secretory mechanism, unlike other parvoviruses, which just simply accumulate in the cell, and then once there are too many capsids, they just lyse the cell, and this is quite devastating to the tissue where these viruses replicate, these lytic exit mechanisms. But ZMBWV doesn't appear to be like that, and this might be that explains why we only see symptoms in animals which are 8 weeks old, even though even the newly hatched larvae are infected, and simply because these viruses, as they replicate inside the midgut wall, aren't as destructive, and it takes a while to eliminate these midgut cells. So to conclude this presentation, we can say that we discovered the infectious agent of a nationwide pandemic, characterized its pathogenesis, and we even found a mechanism that could be a solution to counteract it. ZMBWV is a cryo-EM discovery, which suggests that cryo-EM is indeed a suitable alternative virus discovery method to assess the pathogen behind a pandemic. We could set up a model of the pathogenesis of ZMBWV, suggesting that the virus enters the host via the oral fecal route, then starts replicating in the midgut wall. Then once the midgut wall is destroyed, breaches the midgut, invades the fat bodies, and this is what leads to the ultimate death of the animal. However, if the damage into the midgut wall and the virus titers don't reach this critical high point, then the animals actually survive, manage to pupate and complete this life cycle, and then reproduce, and their offspring carries this virus forward. So we can probably assume that this pandemic is kept alive by these asymptomatic individuals that keep on reproducing. We could also show that CMBWV is indeed distributed nationwide, though with a really high degree of variation in its virulence, and that all virulent strains have a single common origin in North America. And then ultimately we can say that cryo-EM is sufficiently matured to be used as a virus detection as well as discovery method. And with this, I would like to thank for everyone who contributed to this study, as well as a huge thank you goes out to everyone who provided us with samples. And then would like to thank for your attention with my lizard, who's actually enjoying a symptom-free super warm meal on his own.